So this gives us a polar water molecule. Within our NaCl solution, we also have a charge distribution because it's an ionic compound. So NaCl contains 1 plus charge for the sodium metal ion and a 1 minus charge for the non-metal chloride ion. So because we've got positives and negatives in each of the components here, partial positives and partial negatives in the water molecule, and true positive and negative charges in the sodium chloride, then we can actually get an association between these different components. And that's essentially what allows the water molecules to interact with the salt to be able to dissolve it. So what actually happens is the sodium chloride will actually dissociate and separate into its 2 separate ions. So essentially the sodium positive ion will come away from the negative chloride ion. And these ions will actually be stabilized by multiple H2O molecules. Let's have a look exactly how this works. So in the animation here, we can see that we've got several water molecules, and each water molecule has this arrangement of having the partial negative on the oxygen and the partial positives on the hydrogen, which is going to be important for what happens when we're forming this solution. So remember, in the solution we're going to have lots more water molecules than we will salt. So in this example I'm just showing you a fraction of the water molecules present. So the water molecules will associate with the salt and take away 1 ion at a time off that ionic compound, which is the sodium chloride. So we're going to strip the chloride ion off that collection of ions there and stabilize it by allowing the water molecules to associate in a way where the partially positive parts of the hydrogen are pointing towards the negative chloride ion. So you can see there's an attraction between the partial positive hydrogens and the partially negative chloride ions, which is basically able to stabilize the chloride ion even without the association between the chloride and the sodium. So let's look at that once more. So we get the water molecules coming in, bringing the chloride ion away from the rest of the sodium chloride, and we have a stabilization between the negative charge of the chloride ion and the partial positives on the water molecules. So a similar thing can happen with the positive ion of the sodium chloride. So let's have a look at what happens this time. So this time the positive sodium is going to be taken away from the sodium chloride, and it again can be stabilized by the water molecules. But this happens in a bit of a different way to what we saw for the chloride ion, because this time we actually need the partial negatives of the oxygen to actually stabilize the positive charge of the sodium ion. So because the sodium chloride is composed of positive and negative charges, we need to make use of the water molecules having both partial positive and partial negatives. So in this case, where we strip away sodium, it's actually the partial negative oxygens that are going to stabilize the positive sodium. But in the previous slide, we saw that it was the partially positive hydrogens that were stabilizing the negative chloride ion. So you can imagine that all the water molecules in solution are actually able to stabilize the salt that is being added into the water solvent. So once all of the ions have been taken away from the sodium chloride and have separated, what's actually happened is the sodium chloride has dissolved into solution. So in each case, we need multiple water molecules to stabilize the positive or negative charge of the ions that were part of sodium chloride. So we can see in these two representations that we have the partial positives stabilizing the negative charge of chlorine, and here within the water molecules, we've got the partial negatives of the oxygen stabilizing the sodium there. So what do you think would happen once the solution is saturated? If we added in more sodium chloride, why wouldn't the water molecules be able to stabilize the ions within that salt in the way I've shown you here? Well, once the solution is saturated, that means it can't dissolve anymore. So that then must mean that all the water molecules are all already occupied, already stabilizing the ions in this way. So we would already be using all the water molecules to stabilize the chloride ions and the sodium ions. So then if you added in any more of the sodium chloride, essentially it would just stay together as a solid, and it wouldn't be able to be dissolved in the way I've shown you here, where the sodium and chloride ions can actually associate with the water molecules, because there's no water molecules available to do this anymore. So the last topic we're going to have a look at is osmosis. So osmosis basically describes the movement of solvent through a semi-permeable membrane, and this moves from a concentration of that solvent from high to low. So essentially we have solvent moving from high concentration to low concentration through a semi-permeable membrane. So this is quite similar to what I showed you earlier in the session with diffusion, where the gas was moving from high pressure to low pressure environment. The difference for osmosis is this time we're looking at a solution, and it's also movement through a semi-permeable membrane. So the semi-permeable membrane will actually allow the solvent to pass through, but other larger molecules actually won't be able to pass through the membrane. So a good example of a semi-permeable membrane is actually the cell membrane. So each of our cells have one of these membranes that is able to let certain components through and not others. So in the right-hand side representation here, we can see that we've got the movement of water across the semi-permeable membrane. So on the left-hand side of the semi-permeable membrane, there's more solvent represented by these 9 spheres. Whereas on the right-hand side, there's less solvent, as shown by 4 spheres. So what will happen is the solvent will actually move from left to right through that semi-permeable membrane to try and even it up, so there's an equal amount of solvent on both sides of the semi-permeable membrane. So also want to define what diffusion is, which we referred to also for the gas in the previous part of the session. So for diffusion, this allows a substance to move from an area of high concentration to an area of low concentration. However, the key difference to osmosis is there's no movement through a membrane. So this is just movement within a solution or within a particular compartment, not moving through a membrane. So as I showed you earlier in the session, the gases can diffuse from a high pressure or high concentration location to a lower pressure, low concentration location. And this allows the oxygen to move firstly from the atmosphere into our lungs and then throughout the body. We can also have diffusion of solute. So for example, if there's a high concentration of salt in one location, it can then move to a location where there's a lower concentration, and this movement is via diffusion. So as I said, the key difference between osmosis and diffusion is the fact that in osmosis, it's the solvent that's moving rather than the solute, and also the movement of that solvent is through a semi-permeable membrane. So the main solvent of the human body is actually water. So water is actually able to move in and out of our cells, which are a semi-permeable membrane, via osmosis. So if there's more water on one side of the cell than the other, then osmosis can actually occur to try and ensure that there's an equal concentration of solvent inside and outside of the cell. So there is also other ways that water can move in and out of our cells, and one of those is moving through specialized protein channels within the cell membrane. These are called aquaporins. So this will actually allow water to move more quickly in and out of the cell, and it does this to basically make sure it doesn't have to, the water does this to make sure it doesn't have to come into contact with the cell membrane, which is not favorable for the water to contact that because it's actually very non-polar and what we call hydrophobic. So to summarize what we've seen on this slide, water can move in and out of cells via osmosis and also through specialized protein channels called an aquaporin. So a protein channel you can basically just think of as a little gap within the membrane formed by a protein that will actually allow water to move in and out. So it's this special type of channel called an aquaporin that does this. Alright, so we've reached another participation plus question where you can try and put some of this knowledge into practice about how our ionic compound that's polar, salt, is able to dissolve in water and what the consequence of adding more and more salt would be into that solution of water. So make sure you have a go at this one to confirm your understanding of what we've been through in the session. Also a couple of review questions to have a go at, so remember you can access these on that revision quiz. So to summarize what we've seen in this section, we have the solution being a mixture of components. The solute dissolved in the solvent is what is composed of that in that mixture. So the solvent is the component that there's most of in solution, whereas the solute is the one that there's less of. So you can have multiple solutes dissolved in a single solvent. Solubility of a solution is basically a description of how well that solution can dissolve other components. So specifically, it's the maximum amount of solute that can dissolve in a particular volume of solvent. And once a solution has reached the maximum amount it can dissolve, we then call it a saturated solution, and it can't dissolve anymore. So if you add more solute into that saturated solution, the solute will remain undissolved and it won't become part of the solution. So we then looked at how solutions can form, the fact that they have to have matching properties, so polar solvents dissolve polar solutes and non-polar solvents dissolve non-polar solutes, which we'll have a look at more in a later session. So the example that we were having a look at was the fact that the polar water could actually stabilize the individual ions within sodium chloride by stabilizing them using the partial positives and partial negatives within the atoms of the water molecules. We also had a look at osmosis, which was the movement of solvent through a semipermeable membrane from an area of high concentration of solvent to an area of lower concentration of solvent. And we saw that this allows water to move in and out of the body cells via osmosis. So we've only got one more session to go in the chemistry part of the course. So the next session will be looking at acids and bases, which will be a specific type of chemical reaction that is very important to the body and also contributes to the pH of the blood and other locations within the body. So good luck with your review of this session, and I'll see you for our next acids and bases lecture.